So to take a look at this again, we have skeletal muscle tissue, individual skeletal muscle fiber. It can be called a cell or fiber, indicating the length. The stripes going across the width are collectively known as striations. And as described in lecture, the lighter stripes, the lighter bands, I bands, the darker stripes, the A bands. And along the length of these cells, there's multiple nuclei. And here's a great example here, just a single nucleus. And another sample of skeletal muscle is here. So just a different staining treatment, as you notice here. So we're once again looking at skeletal muscle. The striations are really apparent here, the striping. And you can see now this purple stain applied to the nucleus. So just a second sample of skeletal muscle tissue. And we're now going to look at a third example. The difference is now this is a cross section. The previous sample of skeletal muscle was a longitudinal section going along the length of the cell. In this case, these cells, you can imagine they're running out of the screen. They're cut across their width or a cross section. So on the list of terms, that first one listed, I'm actually going to have you cross out. So the epimysium. And you notice in the lab manual it says may not be visible. So we're going to cross out epimysium. So we're going to start with the perimysium. And that's what you see here is this pink tissue. I'll put an arrow right on it. Is the perimysium. And you notice how it's wrapping around a bundle of cells. And that's its structure and placement. The perimysium wrapping around a bundle of cells. And in fact, the name for the bundle of cells, I'm skipping down to the term fascicle. So now this bundle of skeletal muscle fibers wrapped in perimysium, that's the term fascicle. Moving deeper in now, you notice here we can see an individual skeletal muscle fiber. And then you notice surrounding and between the cells, you can see these faint pink fibers, like right about in here, wrapping right around each skeletal muscle fiber or myofiber, myo meaning muscle. That's where we see the endomysium layer of connective tissue. And we'll zoom in for, of course, that detail. The final trait, the nuclei, well, again, they just appear as these dark purple dots. And you notice how they're right near the border of the cell. So that peripheral location near the edge of the cell, the little purple dots representing the multiple nuclei. And zooming in on the cross section. So we have one skeletal muscle fiber. You can see the pink tissue now directly against the cells. That's the endomysium. And a much closer look closer look at any of the nuclei. So a cross section of skeletal muscle. We could zoom out on the cross section of skeletal muscle, the longitudinal section of skeletal muscle, and we had two different staining treatments for longitudinal sections. So next, that leads us to cardiac muscle, the muscle tissue of the heart. And you notice on this slide, we just have this grayscale appearance. It lacks the bright color treatment for our particular sample. Color and shapes are always the big hints for histology. So an individual cardiac muscle cell or a cardiocyte. Well, we can start with the terminology of an intercalated disc. You notice here, the very, very dark lines, intercalated disc. These are at the boundary, at the border of a cardiac muscle cell. So we could look around, and here would be a great example. From one intercalated disc to the next, that would be a good example of a cardiac muscle fiber. The striations, again, the alternating stripes, light and dark. Collectively, again, striations. The nucleus, we could look for the dark staining of these ovals here, the nucleus. Usually one nucleus per cardiac muscle cell. And again, now all around this sample, you can see the very thick, dark, prominent lines. It's a connection from one cardiac muscle cell to its neighbor. And these are all great examples of intercalated discs. And our last type of muscle, we did skeletal muscle, cardiac muscle, and now smooth muscle. And for smooth muscle, again, we're looking at now smooth muscle tissue made of individual smooth muscle fibers or smooth muscle cells. And to try to find an individual one, here would be a good example. One kind of stands out by itself. If I were to sketch it out, they're thinner on the edge, a little thicker in the middle. So these kind of spindle-shaped cells is the description, a smooth muscle fiber. And then you see the prominent staining here in purple. There's only one nucleus per cell. So the nucleus is the one trait that we could see and point out. So there's just a large mass here of multiple smooth muscle cells, and the nucleus is the one key distinction that we could see under the microscope.